can actually visualize all three stages of the mushroom's life cycle in this slide. So here we have straw. It's cut up into small pieces. I'm just going to replay this. So yeah, it's cut into small pieces. It's not in full length. Now we are putting that into a container. Now this has been boiled for 30 to 40 minutes. The objective of this workshop is to teach people, grow mushrooms in their home. So mushrooms as a crop is very unfamiliar to the Indian people, and mushroom cultivation is even still more unfamiliar, I suppose. And mushrooms seem a little alien to consumers, and the cultivation of mushrooms is a very mysterious thing, and they feel that it might be very difficult to do or something, and this is absolutely not true. Growing mushrooms is as easy as growing any other plant, I would say. So yeah, in this workshop, we are going to begin with one of the easier mushrooms that you can grow in your home. And if you just understand the logic behind growing that mushroom, you will pretty much be able to grow any other variety of mushroom as well. Okay, so let's get right into it. So in this webinar, you will learn what a mushroom is, what a fungus is, and the importance and scope of mushroom growing in India. Okay, so in this webinar, you are going to learn what a mushroom is and what a fungus is, and the importance and scope of growing mushrooms in India. And then you will move on and learn the principle of growing mushrooms. And lastly, the procedure for growing oyster mushrooms at home. So, before I start telling you how to grow mushrooms in your home, it is important that you have a strong understanding of what a mushroom is and what a fungus is, so that you have a good foundation to learn the things that will be taught later on. And secondly, we are going to talk a little bit about mushroom growing in the Indian context. Why it is significant for Indians to grow mushrooms or consume mushrooms. Thirdly, the principle of growing mushrooms. So, mushroom growing essentially it has a logic behind it. You could say the essence of growing mushrooms. Just as there is the essence of growing plants is to put the seed and give it water, give it sunlight, and it takes carbon dioxide, and then it grows. Right? That would be the essence behind growing a plant. There's a similar essence behind growing mushrooms as well. And if you understand that essence, then you will be able to grow mushrooms of many varieties. So yeah, we are going to learn that. And lastly, we are going to apply that essence for growing oyster mushrooms at your homes. And now we are choosing oyster mushrooms here because they are the easiest mushrooms to grow. And they are also the second most consumed mushrooms in India. Okay. Moving on. So what is a mushroom? Mushrooms are fruits of an organism called fungus. I'm sure that you've all seen a fungus in your homes or somewhere out there in the environment where anything is rotting. You would have probably seen a fungus over there. Now, not all fungi produce mushrooms. Only some of the organisms belonging to the kingdom of fungi can produce mushrooms. A mushroom is essentially a reproductory structure of the fungus. Not all fungi have the same reproductory structures. Some fungi reproduce through spores, some fungi reproduce through budding, and only some of them, a portion of the organisms under fungi, reproduce through mushrooms. All right. Okay, we have to let in some people. 1 sec. So a mushroom is a fruiting body of an organism called fungus. Guys, okay. So not all fungi reproduce through mushrooms. Some sporulate, some undergo budding, and some reproduce through mushrooms. So a mushroom is a reproductive structure of a fungus. Now we talked about how mushrooms are reproductive structures of fungi, but what are fungi, right? Um, so the answer is there are different kinds of life forms on the planet, and each of them have very distinctive characters that put them into separate divisions. So in this picture, you're seeing the five kingdom classification of life. Um, very recently they've split up this kingdom called Monera into archaea and bacteria. But just for the purpose of this webinar, we are going to consider the five kingdom classification. So these are the five kingdoms of life. Under Monera, we have very ancient beings called archibacteria and bacteria. They are prokaryotic. They are prokaryotic. They are unicellular. They are sometimes motile, sometimes non-motile, and they mostly reproduce asexually. Okay. So the second kingdom is of protists. Now, protists are organisms that are similar to algae, I would say. They are eukaryotic, meaning that they have slightly more developed cellular organelles, and they are also capable of photosynthesis, and they also reproduce sexually and asexually. And the last three kingdoms are macroscopic kingdoms, meaning that they can be visualized with your eyes. And there we have plants, animals, and fungi. So plants, they are eukaryotic, multicellular, they photosynthesize, they are non-motile and sexual. So these are essentially the characters that are significant, that are unique to the respective kingdoms. And since they have such distinctive characters, is what puts them into separate divisions. And similarly, fungi also have such distinctive characters that put them apart from the rest of the kingdoms. So fungi are eukaryotic, they are multicellular, they absorb nutrients from their extracellular environment, which makes them heterotrophic. They are non-motile and they are capable of sexual mode of reproduction. It should also be noticed that a lot of fungi are saprophytic in nature, meaning that they obtain their nutrition from dead and decaying matter. Okay, moving on to the next slide, we are going to have a look at the life cycle of a mushroom. So this is a mature mushroom that you're seeing here. This is the stipe, this is the cap, and underneath the cap it has these radiating structures called gills, and it's from these gills that the spores will be dropped into the environment. So the mushroom will drop spores into the environment, and these spores are going to get carried. They're going to get carried off to far off locations due to wind currents, and they are going to settle on a number of environments, on a number of substrates, and in a number of places. And some of these spores will land on a location where they have the right climate and the right nutrition and everything that they need to basically germinate. So once the spores find a suitable place to germinate, a tubular filamentous growth is going to begin growing out of these spores, and then they are going to eventually grow out to the point where they will appear as a cottony growth, which we identify as mycelium. The mycelium is the vegetative body of the fungus. I suppose you can actually identify the mycelium as the true form of the fungus, and the mushroom is actually just a fruiting body of the organism. Uh, when the mycelium has established itself comfortably in a growing medium, it's going to form white little knot-like structures, which are called the mushroom primordia. They are essentially the the starting stage of the mushroom, and the primordia will grow into a mature mushroom again, hence completing the life cycle. So the mushroom will drop spores. Spores will fly off and germinate in places that are accommodating. After germination, the mycelium is going to arise from the spore, and from the mycelium, mushroom primordia are going to appear, and they will grow into a mature mushroom again. So here we have this nice video of the mushroom's life cycle. You can actually visualize all three stages of the mushroom's life cycle in this slide. Um, so what you're seeing here is a piece of wood right here, and two mushrooms have emerged from that. And the mushrooms are also dropping spores into the environment um, in a very picturesque manner. And so what you should notice is that the fungus would have first colonized this piece of wood right here, and once it has colonized itself, then it is it wants to make more copies of itself. It wants to propagate its own kind. So hence it grows mushrooms, and after the mushroom has matured, it releases spores into the environment, and they will probably fly off and colonize other pieces of wood just like this, and they will grow mushrooms again over there somewhere, hence completing the life cycle. So next we are going to talk briefly about the importance of mushroom cultivation in India. So India is a country of a very high population, and by feeding this high population we generate a lot of agricultural waste. So we grow rice, paddy, I mean rice, wheat, and all these other crops for a very high population. And if you as you already know, any plant body is probably maybe 10%, 20% of it is the actual food, and the rest of it is all agricultural waste. um, and this waste can actually lead to a lot of ecological problems if not managed properly. Farmers are known to burn the stubble, which causes air pollution. And even if they don't burn it, even if they start dumping it, it's going to start a lot of insect and pest problems. So, the managing of this agricultural waste itself is a huge challenge. So mushroom cultivation gives us an excellent opportunity to turn this waste into protein-rich food, and also to revitalize the soil through composting. So mushroom cultivation is something that can be done by utilizing all these agricultural wastes, and once the agricultural waste has been used to grow mushrooms, then again the spent agricultural waste or the spent mushroom substrate can again be composted very easily. So you're utilizing the agricultural waste to grow food, and then you can also compost it to revitalize the soil. So it's a multi-edged sword in our effort to save the ecological balance, you can say. So that is the importance of mushroom cultivation in India. Now we are going to talk a little bit more about the scope of mushroom cultivation in India. So India has a very high population, as I said earlier, which needs to be fed every day. There's also a lot of agricultural waste, and the other thing is that there is also a lot of demand for high nutritional food. And there's a lot of agricultural waste and also demand for high nutrition food. So one fulfills each other. and labor is cheaply available. Mushroom cultivation is requires a lot of labor. It is labor intensive, and India is one such country where labor is cheaply available. Hence, it is something that is some it's a factor that makes mushroom cultivation very pursuable for Indians. Mushroom growing is a sustainable method of growing highly nutritious food for the people who need it. Mushrooms can be grown by farmers with less land too, with integrations of vertical farming practices. So, mushroom is a horticultural crop. Mushroom cultivation can be done indoors, and there is scope for growing more mushrooms per square feet compared to other crops. Mushroom cultivation can provide employment and additional sources of income to farmers while also utilizing cheaply available raw materials. So, since all these factors are since all these factors are playing a role, we have high population, high agricultural waste, labor is cheaply available. So doing mushroom cultivation will also generate jobs for rural farmers. So that is the scope of doing mushroom cultivation in India. Uh, Next, we are going to talk about the general principle for mushroom growing. Okay, until now we have talked about the basics of mushroom cultivation, the scope, the importance, and everything. And now we are going to get down to the actual process of growing mushrooms. And from here on is where you get your value out of this webinar. So please pay attention. So I believe that there are 4 steps: preparation of the substrate, spawning, incubation, and fruiting. So if you just understand the logic behind each of these steps, you will be able to grow any kind of mushrooms. So, I suppose a lot of you all who are attending this webinar are from a scientific background. You've probably done some inoculations in the lab. You're from biotech and microbiology backgrounds. So, what do we do when we are inoculating a microbial culture in the lab? So first we essentially, sterilize a nutrient medium in the autoclave, and then we inoculate it in the laminar airflow. We incubate it in the incubator, and then we allow it to grow out, right? So that is exactly what we are doing here. The fungus is also a microbial culture. It is a microbial culture, except that we are doing this on a not so much of a laboratory setting. That's all. So these are the 4 steps that you will need to understand. If you understand these 4 steps, you will understand the essence of mushroom growing. So the first step of this general principle is the preparation of the substrate. So the substrate serves as food material to grow the fungus. The substrate can be anything like straw, corn cobs, bagasse, sawdust, cardboard, soybean hulls, cotton seed hulls, paper. So you see, they are all dead plant materials, dead and dry plant materials that we can use as substrate. Just as how you are using a nutrient media to grow microbial cultures, you are using the dead plant materials as a substrate to grow the fungus. So the substrate is wetted and subjected to heat treatment. Firstly, I'm just letting in some more people. Okay. So in the first step, the substrate is going to be wetted and it's going to be subjected to heat treatment. In nature, all these substrates, they grow out in nature. They will have a lot of microbes on them. So we will have to reduce the microbial load on these substrates first before we begin inoculating these substrates with the fungus. So that is the that is the objective of doing heat treatment on the substrates. This will eliminate all the microorganisms that would compete. Okay, so these are the two methods that is employed in mushroom cultivation for the heat treatment of the substrate. And we do the heat treatment to kill the microorganisms that are infesting our substrate. So the second step would be spawning. So the second step is spawning. In this step, we are going to mix the pasteurized substrate and the inoculum, which is the spawn, together. So during spawning, the pasteurized or sterilized substrate is mixed with the mushroom spawn, which is the medium that is carrying the fungus. Okay, a spawn can be many things. It can be grain spawn, it can be in the form of a spore syringe or a liquid culture. But for most commercial mushroom cultivating operations, it is in the form of grain spawn, which you can see in the image here. So you are seeing that this lady has pasteurized some straw over here, and she has a plastic cover, which is the container, and she is loading the pasteurized straw and mixing that with a little bit of spawn right here, and then she is going to close this off. This process is essentially is what spawning is. So something else that you must know: after pasteurization or sterilization, the substrate must be handled very hygienically. It should not come in contact with dirty surfaces or hands. You just pasteurize the substrate to eliminate all the microorganisms, and right after you pasteurize it, if you are going to take it out of the pasteurizer and keep it on a dirty surface or touch it with dirty hands, that is actually going to be counterproductive. You are going to add more microbes in there, which may harm our fungus. So that should not be done. Aseptic techniques must be maintained. If you're going to keep the straw on a surface, the surface should be sanitized. If you're going to touch it with your hands should be sanitized. And when you are mixing the straw and the spawn together, if your hands will have a lot of contaminants, your clothes will have a lot of contaminants, your hair will have a lot of contaminants. So yeah, once you sanitize your hands, only after that you should touch the spawn and the substrate. And if you have to, if you are compelled to scratch your head or touch your clothes for any reason, then you must re-sterilize your hands before touching the substrate and the spawn. So yeah, aseptic techniques must be maintained, and it should be done very hygienically. Otherwise, your grow bags are going to develop contamination inside. So after sanitizing our hands, we must add the pasteurized or sterilized substrate and the spawn into a container in appropriate ratios. So how much substrate to add and how much spawn to add, that is something specific to the mushroom that is being grown. We are not talking about any mushroom in particular yet. This is just a step for all the cultivation of all mushrooms, the spawning step. The container must be closed off to prevent entry of dust and insects. So once the substrate and spawn have been mixed together, the container must be closed off, and and then it will be taken for incubation. So this is a picture of a setup that we had done in my city. This is actually my own setup, which I had a little while ago. So the third step is incubation. So incubation is a very easy step. You don't have to do anything here. The substrate has been pasteurized, the fungus has been inoculated, and now all you have to do is put that into a room which can facilitate the fungus to colonize the substrate or eat the substrate. Yeah. And by facilitate, what I mean is just keep it in a temperature that is suitable for the growth of fungus. That is all you need to do. In this step, we will be providing optimum growing conditions for the mycelium and encouraging it to completely colonize our substrate. The prescribed optimum temperature needs to be maintained during spawn. That is all you need to do. And during incubation, you should not place your bags in direct sunlight or anything. It should be kept in a cool and shaded area. So the fourth step is fruiting. So you can probably see in the image here, all the straw inside the bag has turned white, which means that the fungus has colonized that straw. So fruiting is initiated by introducing the colonized substrate to fresh air and humidity. So the fungus was in colonization mode all this time, and once the substrate has been fully colonized, we are going to give it certain signals so that it thinks that to basically signal it that to say to it that we are giving you conditions to grow mushrooms now. We are signaling it to grow out the mushrooms. Yeah, and these signals are to provide fresh air and humidity. So fresh air, to provide fresh air, all you need to do is open one or two windows inside the growing room. That is all you need to do for home growing at least. And to increase the humidity, you need to spray water inside the growing room. So during incubation, the last step, you are only controlling the temperature, right? So in the next phase, which is the fruiting, you will need to control three parameters, which are temperature, humidity, and fresh air. Yeah. And before you were controlling temperature, and in this step, you just introduced air exchange and humidity. And this air exchange and humidity are the triggers for the are signals for the fungus to start growing mushrooms. Okay, guys. So those were the general steps for growing all mushrooms. So no matter which mushroom you are trying to grow, these four steps will be common. Preparation of the substrate, where you provide, where you give heat treatment to the substrate, and then spawning, where you add the spawn and the substrate together. And the next step is incubation. You set aside that container in a comfortable temperature. And next is fruiting, where you introduce humidity and fresh air exchange to make the mushrooms grow out. So yeah, no matter which mushroom you're trying to grow, these steps will be there. And lastly, we are going to talk about, growing oyster mushrooms in your home using plastic buckets. Now, I'm going to teach you how to do this in old plastic buckets, but keep in mind that you can switch the bucket with a plastic cover anytime and follow the instructions in a similar manner and achieve the same result. So, the requirements for growing oyster mushrooms in your home with the bucket tech are: firstly, you will need a bucket with 0.5 to 0.7 inch holes and a lid. Now the number of holes, it's quite flexible, anywhere around 5 to 10 holes you can use, which should be equidistant. Um, they should be placed in a equidistant manner. And you are going to require straw, because I'm recommending straw because straw as a substrate is very easy to handle. It has easy availability, and it is very easy to pasteurize as well. If you were to use sawdust, then if you were to use sawdust, you would need to sterilize it. People may not have the resources to do sterilization, or you can't use your pressure cooker to do sterilization for large amounts of substrate. So yeah, so for substrate you can use paddy straw, wheat straw, or ragi straw. And thirdly, you will need mushroom spawn. Mushroom spawn is essentially grains which are carrying the fungus on them. Yeah. And fourth, you will need a sanitizer. Any hand sanitizer is probably 70% alcohol. So you can also use, if isopropyl alcohol is available to you, then you can use isopropyl alcohol or a hand sanitizer. or or in the worst of circumstances, you can use mild Dettol solution. But I recommend you using a hand sanitizer for the best results. And fifth, you will need a micropore tape. This micropore tape you can find at any medical shop near you. Just ask for micropore tape. These tapes they allow gas exchange to occur. And lastly, you will need a small water sprayer, which you can also buy locally very easily. So these are the requirements: a bucket with uh, half to 0.75-inch holes, and a lid. A lid is also a necessary thing, guys. So make sure that you get a bucket with a lid. If you don't have one, you can probably buy one at your local scrapyard for like under 50 rupees. Yeah. And then paddy straw, wheat straw, or ragi straw, one of these, mushroom spawn, sanitizer, micropore tape, and a water sprayer. These are the things you need to grow mushrooms in your home. Okay. So I have prepared video footage here of how these steps should be performed. So first step was preparation of the substrate, guys, right? I told you earlier. So the straw is cut into small pieces and submerged in water and boiled for 40 minutes. And that does our pasteurization. After pasteurizing the straw, excess water is allowed to strain out over time until optimum moisture is retained in the straw. So, I'll just show this to you how this is done on video. So here we have straw. It's cut up into small pieces. I'm just going to replay this. So yeah, it's cut into small pieces. It's not in full length. Now we are putting that into a container, and we are going to weigh it down by placing something heavy on it. And now I have completely submerged that plastic bag with water, and now I'll begin to boil it. And meanwhile, the bucket is also ready. Now this has been boiled for 30 to 40 minutes. So here we have straw. It's cut up into small pieces. I'm just going to replay this. So yeah, it's cut into small pieces. It's not in full length. Now we are putting that into a container. So keep in mind the surface that I'm trying to keep the substrate on, the pasteurized substrate on, that is very clean. That grill mesh has also been sanitized actually, so that no microbes have the opportunity to enter the substrate after pasteurization. Now this has been boiled for 30 to 40 minutes, right? Next, we are going to prepare the bucket for spawning. The bucket and the lid should be thoroughly sanitized, and all holes should be covered by a micropore tape. Okay, if you just have a look inside this bucket right here, it's sparkling white. Everything has been cleaned. It was washed once with soap and then sanitized with a hand sanitizer. Yeah, and now we are going to cover all these holes with a micropore tape. Okay, and once we have covered all the holes with a micropore tape, and then we are going to add the pasteurized straw and mushroom spawn into the bucket as shown in the video. Now, what am I trying to do here is, I'm squeezing the substrate to make sure that there is no excess moisture present inside the substrate. Now, if pasteurization is done as I just showed you, then you must actually let it rest for around 3 to 4 hours to allow all the excess moisture to go out. So when I squeeze, and if it's okay if one or two drops of water come out of the straw and hit the ground, but if it is dripping a lot, if it is very drippy, then that means that the substrate is very wet, and you should probably strain it for a longer time. So when you squeeze the substrate in the palm of your hand, your palm should get wet, but water should not be, a lot of water should not be dripping from it. So the first layer is always of the pasteurized straw. We will add a 3-inch layer of pasteurized straw and press it down as tightly as we can. Okay, I'm just going to replay this. So this is mushroom spawn. This is mushroom grain spawn right here in my hand. So we are sandwiching the mushroom spawn with layers of pasteurized substrate. First we'll add substrate, then spawn, substrate, spawn, substrate, spawn. And the amount of grain spawn, it should roughly be a handful. That's all. A handful comes close to 10 grams. More or less will not affect the cultivation process significantly. So it's all right. So, after the box has been adequately filled, we are going to close it off with a lid, which has also been cleaned with a sanitizer. Remember that. Okay, I'm just going to replay the whole thing again once later, but for now, let's move on. So, incubation, as I told you, there's not a lot to be done in the incubation. Once I closed the lid off, I just kept it in a room where it has optimum temperature for fungal growth. So I let it sit there undisturbed in the temperature range of 22 to 27 degrees for roughly 15 to 20 days in a cool shaded area. And after 15 to 20 days, I slightly opened the lid to see if all of the straw has been colonized by the fungus. So when I opened the lid, this is what I saw. The substrate was completely covered by the fungus, and incubation was a success. The fungus has colonized all the substrate. Next comes the fruiting part. So to fruit the substrate, to fruit the colonized substrate, what we need to do is we need to remove all the micropore tapes off the bucket. We had covered all the holes with the micropore tape. We are going to take that off. And during incubation, we just kept it in a room that had an optimum temperature. That is all we did. But after taking the micropore tape off, we must also increase humidity in the growing room and give it fresh air exchange by opening any windows that are in the grow room. So first thing we're going to do is remove the micropore tapes. Secondly, we'll spray water inside the grow room to increase humidity, and we will open windows inside the grow room to provide ventilation. Spraying of water and opening of the windows, these were all not necessary during the incubation phase, but during the fruiting phase, they will be necessary. As I told you, they become the signals for mushroom growth. So humidity should be provided by spraying water indirectly. Okay, so we need to increase humidity of the air. That doesn't mean that we should spray water on the mushrooms themselves. The water should be sprayed like on the ground or into the air, but not directly on the mushrooms. Just try to spray the water indirectly, like just spray water around the bucket rather than directly on it. That's all. It's okay if a little bit of water touches the mushroom, but just don't like water the mushrooms. Yeah, so those are the 4 steps you need to do to grow oyster mushrooms in buckets in your home. So I'm just going to have a quick rundown of all the steps, just the video, so that you guys understand it properly. We took cut straw, submerged it in water. We gave it heat treatment for around 40 minutes at a temperature range of maybe 70 to 80 degrees. Then we took the substrate out, kept it on a surface that is clean, that has been cleaned with a sanitizer. We should treat the pasteurized substrate as we are treating food. We only keep our food on clean plates, right? That's how it is. Okay, that was preparation of the substrate. Now preparation of the bucket. Remember to clean the bucket with sanitizer, right? We should clean the plate as well, right? So we should remember to clean all the inner surfaces and maybe the outer surfaces as well. First wash it with some soap water and then sanitize with an isopropyl alcohol or a hand sanitizer for just to be extra sure. And this is also a very important step, guys. If you do not clean your bucket thoroughly as possible, then there is a huge risk of contamination and your grow might end up failing. So after cleaning the bucket, we are going to put the micropore tape on all the holes. Then after making sure that the straw is strained of all the excess water, you should let it strain for 3 to 4 hours. Because if you just pasteurize your straw like I did by submerging it in water, then it's going to have a lot of excess water. So you should also let it strain for some time, maybe 3 to 4 hours, so that when you squeeze the straw, um, like this, water is not dripping down, right? Yeah, the straw is wet, but it's not dripping wet. So the first layer is of the straw. So the first and last layer will be of the straw, and the spawn will always be sandwiched. The first layer will be of straw, and the last layer on top will also be of the straw. As I perform these steps, my hands have been sanitized. I would not touch the pasteurized substrate with dirty hands. So yeah, that was spawning. And then I just took this bucket and kept it in a room where optimum temperature is there, away from direct sunlight, so that the fungus has time to colonize the pasteurized substrate. After 15 to 20 days of incubation, the fungus has completely covered the substrate. And then I removed the micropore tapes off. I opened the windows, I started spraying water, and this is how the mushrooms started to grow out. See, I'm not spraying water on the mushrooms, I'm just spraying water in and around the bucket. This is day 2 of the fruiting. They will double in size every day. So once the pinheads appear, they will reach maturity in probably like 4 to 5 days. This is day 3. See, there's a lot of difference in size just from day 2 and day 3. More spraying. And this is on day 4 when they are ready to harvest. Okay. And then the final step of harvesting. So we must hold the mushrooms at the base and give them a twist to remove the clusters from the bucket. Watch how I'm doing it. I'm holding them at the base and giving them a twist. Yeah. So that's how you do it. So the mushrooms have a shelf life of 2 days in room temperature and 4 days if refrigerated. Oyster mushrooms taste best when it is stir fried or in sautéed dishes. So they don't. I mean, in my opinion, they don't taste very great in curries. So in dry items, in stir fries and sautéed dishes, they taste best. So yeah, that's how you grow oyster mushrooms in your home. So lastly, I just want to add this. I wanted to add this slide to tell you all what Fungipedia is. So Fungipedia is an educative online portal. We want to connect the Indian people with mushrooms. We educate people about the art of mushroom growing and the wonderful benefits that fungi and mushrooms have to offer. We hope by doing this we can eliminate consumer resistance among Indians regarding the use of mushrooms in their diet. We also sell mushroom growing kits, mushroom spawn, and we are also looking forward to making value added products with mushrooms that are locally grown. I'm just going to take a second and let you guys see this slide. So this is a WhatsApp group that I've made, which has a lot of which has many mushroom growers in there. So when you actually begin to grow mushrooms, you might end up facing a few problems or having a few doubts. If you just join this WhatsApp group that I've created, and there are a lot of mushroom growers in there, a lot of veteran mushroom growers, and myself. If you whatever doubts you have, whatever queries you have regarding mushroom cultivation, you can just ask us there, and it will be answered by experienced growers. And you can also contact me on my phone number anytime you want. If you want to purchase mushroom spawn, I actually own a lab which manufactures mushroom spawn. You can contact me anytime you want, and I can send the mushroom spawn to your address. So yeah, so thank you for joining us on this webinar. I made sure to keep it short and sweet. I hope you guys liked it. And now we have time for a Q&A session so that if you have any doubts, they can be clear. So guys, you can ask me anything you want, and there are no wrong questions. So just feel free to shoot. Okay, I see some questions in the chat. Okay. Please explain how to manage contamination in buckets. So yeah, one disadvantage of doing bucket tech is that you will not have a visual of what's happening inside the bucket. So the only way to avoid this is to do your steps right. Do proper pasteurization. Don't cut corners. Extend the pasteurization by 10 minutes if you want, but don't make it less than the prescribed amount of time. And make sure that the right temperature is reached, and it gets the heat treatment for the right amount of time. Make sure that that happens. And once the pasteurization is done, post pasteurization, make sure that it's not handled in dirty with dirty hands, or it is not kept on dirty surfaces. And even after sanitizing your hands, if you're compelled to touch your you know your if you're compelled to scratch your head or touch your clothes or something, there are already maybe thousands of microorganisms there. So if for any reason you must touch dirty surfaces with your hand, then make sure you re-sterilize your hand. As you are doing the spawning, if you just scratch your head once, your fingers will catch hundreds of microorganisms. So if you touch anything else other than already clean substance surfaces, then re-sterilize. So re-sterilize your hands, keep them really really clean. So yeah, pasteurize well and do the spawning clean. And oyster mushrooms are quite strong. If you just do those two steps in a very efficient manner, then contamination should not be an issue. Yeah. Okay, how to increase vitamin D in mushrooms? Well, as far as I know, mushrooms when you expose them to sunlight, post harvesting, also you can increase the vitamin D amount in them. Yeah, it's as easy as that. You just expose them to UV light, and they will develop vitamin D. They will. I think I think they turn ergosterol into vitamin D. I'm not sure about the chemical nuances. My specialty is just in cultivation. Um, yeah, so yeah, you just expose the mushroom fruiting bodies to UV light, and they will develop vitamin D on their own. Yeah. To sterilize or pasteurize at home, do we need to buy these containers? Okay. So to pasteurize the substrate, what you can do is you can get I mean, there are a lot of things you can improvise with. The only condition that you need to fulfill is that the required temperature is reached for the required duration of time. So what I would suggest is, if you are pasteurizing a lot of substrate, one, you can use a container as I showed you in the video, or if you are pasteurizing a lot of substrate, then what you can do is you can buy a 55-gallon drum of the heavy gauge. You can also find that in the scrapyard. They will be used to carry oils for petroleum companies. You can use that as a large container. You can put your substrate in, fill it with water, and you can boil that. And for sterilization, for very small amounts of substrate, you can use the pressure cooker that is inside your home and sterilize it for 60 to 90 minutes. But again, if you are sterilizing a lot of substrate, then you would need to buy an autoclave. You can buy a portable autoclave which is smaller, which is roughly 1.5 to 2 feet in, it's just this tall. And if it's even still more amount of substrate needs to be sterilized, then you need to buy an industrial autoclave. Yeah. So how many times we can reharvest after first harvest? Oh wow. Okay, I'm sorry. I think I should have spoken about this as well. So yeah, mushrooms will fruit from the container about 3 to 4 times, depending on a lot of factors. Yeah, so once you harvest the mushrooms, just provide even after uh, you harvested the mushrooms, just keep them in the fruiting phase. Keep spraying water, keep giving them fresh air, and they will just fruit again. Yeah. So you can harvest. So until you take out some 3 to 4 harvests, you can just keep them in the fruiting phase. Keep them in the fruiting phase. Matlab, just keep spraying water. Just keep providing them with ventilation. That's all, and they will keep fruiting multiple times. If we want to grow oyster mushroom in brown rice, can we grow? And what are the precautions we have to take? Okay. Okay. So brown rice is a nutritionally rich medium. So for a medium like brown rice, you will need to sterilize it. So that is the only, yeah, you will need to sterilize it. That is the only precaution that you will need to be careful about. And probably while inoculating also, probably while inoculating also, you will need to be careful because these brown substrates like brown rice are rich in nutrition. So anything that has a lot of nutrition will also attract a lot of contamination. So yeah, just do sterilization. Don't pasteurize it, sterilize it, and do inoculation in a very very clean manner. Yeah. Pasteurization with waste decomposter, is it a good idea and what are the chances of contamination? To be honest, yeah, I haven't tried this myself. I hear a lot of farmers are trying this. Um, I cannot give you good feedback on this, but yeah, I heard about people using it though. Yeah, I suppose this is like pasteurization by microbes. Like you saturate a medium with microbes that are not hostile to our desired fungus, but they are hostile towards contaminations. So essentially, you are treating the substrate with microorganisms that are friends to our fungus and enemies to the enemies of our fungus. I hope that makes sense. So yeah.
Deepak, can I ask you a question?
Yes, sir. Yes, sir. Hi, Vile, sir. So nice of you to
Hi,
join.
Deepak.
Uh,
Yeah, thanks for the wonderful talk, Deepak, and you have explained it very nicely.
Thank you.
My question is probably I might have asked you the same question before, but
Okay.
for this benefit of this audience, I would like to ask the same question: is mushroom cultivation and selling mushrooms in the market
Mhm.
is a profitable business?
See, cultivation of mushrooms is something that can be done by everyone. But the selling of mushrooms, yes, yeah, it's actually quite doable. But one needs to not only learn about cultivation, but also the other things of about how their local market work, and it is very region specific. For example, someone in Bangalore might have a very easy time selling mushrooms than compared to a village somewhere far outside. But it's not, I don't think it would be right for me to speak on all of these prospective growers. But yeah, I mean, with the right kind of effort and the right kind of knowledge, more importantly, it is definitely possible to sell the mushrooms. Yeah, yeah, it is definitely possible to sell the mushrooms. It's just a matter of being aware of a lot of techniques and methods of, in these days you should learn things like branding and yeah, having the right market placements, having the finding the right distributors. Yeah, it's more about a market knowledge than a cultivation knowledge when it comes to selling of the produce. So that is another discipline that you have to learn, and that also is a very long road. I don't want to like push people into cultivation like that, but yeah, but on the other hand, it is definitely possible. Yeah.
My point is it profitable? Like if I
Oh yes,
invest one
definitely
crore,
profitable. Yes, it's definitely profitable. Like you're turning agri wastes into nutritious food. So yeah, it's a low investment operation with a chance of yielding a lot of profit. Yeah.
what would be how much would be the return on investment?
I mean it is very relative. Matlab, it would depend on a lot of factors like how cheaply you get the substrate, how efficiently you can do it, how many labors you would need, whether you would do the work yourself. Yeah, but since your question needs some kind of satisfactory answer, I would say maybe the return would be like 4 to 5 times of the money you put in.
Oh,
Yeah.
that's wonderful. Thank you very much. Thank you so much. Yeah.
Thank you, sir. Any other questions? Living in Delhi, where can I get ragi straw from or normal straw? Also, can we reuse straw for another batch of mushrooms? No, you cannot reuse the straw for another batch of mushrooms. You can, however, compost them easily. Um, I'm not sure about sourcing raw materials around Delhi, but I think Delhi has a lot of wheat growers in the vicinity. So wheat straw is also does just as well as ragi straw or paddy straw. Visit any? Okay. Is it recorded and can be shared? Yes, I have my good friend Manohara who is in the webinar. He's helping me with recording the session, and it will be uploaded on YouTube. Do we need to buy mushroom spawn after every harvest or can get them from homegrown mushroom? You can actually make the mushroom spawn yourself, but it's a little bit of a lab heavy work, and yeah, you could provided you know how to, but that is another uh, thing that you'll need to learn in depth. Yeah, it's possible, but yeah, you'll need to learn it, and it's quite difficult, not doable by the average person. As I'm teaching oyster mushroom cultivation, as I'm claiming anyone can do it, but spawn production is has a much more difficult barrier. Yeah. Does indirect sunlight require during fruiting stage in Rome? Yes. So mushrooms do require a certain amount of light, but I wouldn't worry about it. During daytime you get a certain amount of ambient light inside the growing room, and that itself will be sufficient for mushroom growth. Yeah. And this is for home growing. Yeah. And I'm sure that all homes get a little bit of sunlight in the day, and that should be enough. Will it be same procedure for button mushroom? Why casing procedure is required? Okay. So the procedure is kind of same. You have a substrate and then it undergoes pasteurization. Not pasture. The pasteurization is a little bit different for button mushrooms. It involves a little bit of what aerobic. Uh, it is just treated aerobically so that the microbes themselves produce the pasteurization temperature, and yeah, it's a little bit different. The pasteurization procedure is different, but pasteurization is done nonetheless. And why is casing procedure required? So certain mushrooms require a little bit of support to grow on. So casing is essentially a medium that provides support for the mushrooms to grow. Yeah, some mushrooms require that to be able to grow upwards. Yeah. Do you know any spawn suppliers in Kathmandu? Uh, I'm not sure, but I'm sure there are plenty of spawn suppliers throughout India. Just try to Google it. You will find it. You'll find one. Can we get participation certificate? Well, I have not made any preparations for this, but if I don't know, it can be done. Yeah, I'll need to think about it. Is it possible to cultivate milky mushrooms in this bucket method? Yeah, it's definitely possible. Yeah, it can be done. But yeah, the casing step needs to be done, which I did not explain here. But if you know how to do casing, then yeah, bucket tech can also be done with milky mushrooms. During the vegetative stage, is it necessary to keep the bags in the dark? Um, Well, it's not really necessary, but I think it would have. I'm sure it has a role to play, but it's definitely not necessary, because if you're doing the bucket tech, it gets no light and it still does well. And if you're using plastic covers, then it definitely gets some light and it still does well. So yeah, just for cultivating in small scales, I won't worry too much about light as a factor. Do you buy or sell mushrooms? So we sell mushroom spawn, we sell mushroom growing kits. We also plans of buying mushrooms, but that would be from people who are very close to us, close to Mysore in Karnataka. Yeah. What is Darshan sir is asking? What is the additional advantages of using bucket? Any special advantages? Okay, so the advantages of using a bucket is, one thing is it can yield a lot compared to plastic covers. It will give a lot of yield. When I tried to grow in that bucket, that almost yielded me 1.5 kilos of mushrooms. And the other thing is, plastic covers will need to lead to a lot of environmental pollution, but this bucket can be reused again. So that is the main advantage, I suppose. Do you give training on mushroom cultivation in offline? Well, I used to have a mushroom cultivation setup, which is not functioning right now. Right now, I'm doing mushroom spawn production in my laboratory full time, and I don't really get any time to manage any cultivation setup as of now. Yeah, so no offline cultivation programs. Any oyster mushroom buyers you know? Yes, I know quite a few of them, but firstly, yeah, well, it's best if you are inside Karnataka, then it would make sense for me to put you in touch with them. But if you have dry mushrooms to sell, then yes, please get in touch with me. I cannot give you a price guarantee or anything, but there are definitely buyers. Yeah, I can put you in touch with them. Any other questions, guys? Feel free to ask me any questions. You're welcome, Persley. Thank you. Thank you, Vile sir. How many times can we harvest mushrooms in same straw? So yeah, you can get up to 3 to 4 harvests if you use straw to grow oyster mushrooms. Yeah, I mean it's relative. It depends on a lot of factors, but yeah, roughly 2, 3, 4 times you can harvest. Yeah, you're welcome, guys. You're welcome. Thank you for attending. Yeah. Substrate to spawn ratio. Okay, good question. So, substrate to spawn ratio, if you want to get down to numbers, then it should be 5 to 7% for oyster mushrooms and straw. Any other questions, guys? How do we know if we can reuse the straw after a harvest? We cannot. We cannot reuse the same substrate after a harvest. Um, the only thing we can do is put it into composting. Yeah. Is there a minimum amount of layers, like if we have a smaller bucket? There is no minimum amount of layers, really. Yeah, but there is. Yeah, I mean, yeah, there's no minimum amount of layers or anything. Yeah, but yeah, I mean, it should just make a compact. It should just be big enough to be a compact structure. That's all. Okay, we will be ending the webinar very soon. So if anyone has any questions, no matter what, please do ask. Okay, I think. Thank you, everyone. Thank you for joining in and making this session worthwhile for me and everyone else. Thank you for your amazing questions. Any frequently asked questions, nothing is coming to my mind right now. Can we microwave the straw while in water? I'm not sure really. I haven't tried it. What you can do is steaming is another popular option that you can try. Steaming actually takes the temperature higher, like up to 90 to 95 degrees it will take. I mean, it's not even very difficult. You just need to make some minor tweaks in the same pasteurization method. There are a lot of videos on YouTube that you can watch how to make a small steaming setup for pasteurization. Yeah. Okay, guys. I'm just going to show you this slide again. Yeah. So you can just scan this QR code and join my mushroom cultivation group. And any queries you have further on, and you can also interact with a lot of mushroom growers who are already on the group. So just if you need to buy mushrooms, want to grow mushrooms, then you can contact me on this number anytime. Okay, thank you all. So yeah, I'll be ending the webinar now. Once again, thank you all for joining.